Good afternoon and welcome everyone to the ICTS Precision Medicine session at Venture Café. I'm your host, Rob Culverhouse. I'm from the Department of Medicine at WashU. We meet on the third Thursday of the month at 4 p.m. and our sessions run about 50 minutes. Our mission is to connect innovative biomedical science generated by WashU, SLU, and MU with entrepreneurs and businesses in the St. Louis region. We hope these sessions stimulate your thinking, and we encourage you to share your insights and ideas with the speakers after their presentations. In case you'd like to follow up with either of today's speakers, you can find their contact information on our website, the ictspm.ustal.edu, and I hope Tricia should post that web address in the chat box. So please feel free to unmute yourself at any time you have that you have questions. And you can also use the chat function. Today's presenters are from the WashU School of Medicine, Chris Posilla and Aadel Chaudhuri, both from the Department of Radiation Oncology, will tell us about their novel methods for obtaining cancer biopsies. Chris Posilla is a graduate research assistant in the lab of Chen and the chief technology officer of their startup. He was named top emerging investigator for 2020 from the Imaging Elevated Symposium, and the lab's current active research includes improving brain imaging using ultrasound and ultrasound-mediated brain drug delivery. The mission of the lab is to develop innovative, focused ultrasound methods to help patients with brain diseases. Aadel Chaudhuri received his Ph.D. in biology from Caltech and his M.D. from Stanford. He is now an assistant professor at WashU. He's received many awards, including Young Investigator Awards from the American Society of Clinical Oncology and the Cancer Research Foundation. His research focuses on the identification and measurement of circulating tumor DNA as a cancer biomarker. He and his colleagues have demonstrated that measuring this after cancer treatment is a biomarker for disease relapse and can also help identify effective precision medicine treatments. So thank you both for being with us today. Chris will start us off telling us about the Hong Lab's development of a technique to use ultrasound as part of a non-invasive system for molecular diagnosis of brain tumors. So welcome, Chris, and take it away.
Great. Thank you so much for the kind introduction. I'm really looking forward to our time today. As Rob mentioned, my name is Christopher Passia, and it's my honor to be on your screens to share some information about our Sauna biopsy technology. So in a nutshell, Sauna biopsy enables non-invasive brain biopsy using a focused ultrasound device. But I'd like to first start with why we need brain biopsies. Brain cancers are widespread and costly to both patients, clinicians, hospitals, and payers. And over 700,000 Americans live with a primary brain tumor, and nearly 90,000 new patients are diagnosed each year. On top of that, about 200,000 patients are diagnosed with brain metastasis each year. And the treatment of brain diseases has been improving over the past decade through precision medicine, which is a technique that requires a diagnosis of the tumor's genetic profile. Now, the standard diagnostic procedure for brain tumors is magnetic resonance imaging, or MRI, where an image of the brain is taken to evaluate the presence of a tumor. However, this lacks the molecular level information necessary for precision medicine. Surgical biopsies can provide this molecular information by extracting a piece of the tumor, but the surgery is invasive and risky. So for this reason, surgical biopsies cannot be performed longitudinally or if the tumor is in an inoperable location. So there are no current diagnostic techniques that offer a safe method to sample molecular information. We present an approach to achieve non-invasive brain biopsies. With sauna biopsy, clinicians can reduce necessary procedures and the risks involved. But because sauna biopsy is a non-invasive approach to patient care, this opens up many new opportunities to optimize treatment and provide better patient care through precision medicine. During the current state of art for surgical biopsy, MR images are acquired to assess the tumor location and be used for neuronavigation guidance in the operating room. Then a hole is drilled into the skull so a biopsy needle can be inserted into the brain to take out a tumor sample. And because this procedure is very invasive, the patient will have to stay in the recovery ICU for a couple of days. Sauna biopsy requires the same pre-surgical MRI and uses the same neuronavigation setup that clinicians are familiar with. This makes it straightforward for doctors to replace the invasive biopsy needle with a non-invasive ultrasound device. And the main value for non-invasive sauna biopsy is the ability to be performed multiple times in a procedure room where a simple blood sample is drawn and the patient can go home the same day. The blood sample will then be analyzed by a third-party company that has an FDA-approved liquid biopsy test for solid tumors. And here is a short video illustrating our technique. The surgical needle is replaced with a non-invasive focused ultrasound device. Focused ultrasound with microbubbles is known to be a safe, non-invasive, spatially targeted technique to reversibly disrupt the blood-brain barrier. And this has conventionally been used to deliver chemotherapeutic drugs, but we are the first to leverage this blood-brain barrier disruption to release molecular biomarkers into the bloodstream. So after focused ultrasound sonication, a blood sample can be taken and analyzed by conventional off-the-shelf methods. And we've already demonstrated that sauna biopsy is feasible and safe in both mice and pig models. In the mouse model, we targeted tumor that's implanted in the brain and observed an increase in the tumor RNA. And in the pig model, we measured an increase in the brain-specific biomarkers that are circulating in the blood. So because sauna biopsy is non-invasive, it can be performed multiple times across the same patient cycle of care. The first application will be to characterize the molecular profile during the initial diagnosis. At this stage, sauna biopsy lowers the risks and costs normally associated with surgical biopsies. Then the second application is enabled by sauna biopsy's ability to be used longitudinally. So neuro-oncologists can monitor the tumor during treatment to tailor their treatment regimen based on what's most effective for the patient. And the frequency of sauna biopsy can be dynamically adjusted based on the patient's needs. So for example, if they have any pre-existing conditions or genetic predisposition. For example, sauna biopsy can be performed at least once a week during a 6-week radiation therapy and at least once a month during the 6-month chemotherapy. And then the third application is motivated by the difficulty in differentiating tumor recurrence from pseudoprogression or any treatment effects. So sauna biopsy can be performed once every 6 months for the patient's entire lifetime to identify the recurrence of any brain tumors without the need for surgery. We've filed and issued 3 patents, and we plan to file more as the technology is further developed. Then our competition can be divided into the gold standards and emerging technologies of focused ultrasound companies that are developing their own clinical systems. The first key attribute is whether the system can provide molecular information. Only surgical biopsy and sauna biopsy offer this depth of knowledge because we're actually analyzing patient samples. However, surgical biopsy is invasive. And this determines not only the safety risks involved, but also the cost of care. That includes the operating room, personnel, anesthesia, the recovery ICU. And lastly, sauna biopsy can be used longitudinally to accurately monitor tumor progression, treatment response, and tumor recurrence over time. And specifically, our entry market includes 1,200 hospitals that have a dedicated neurology and neurosurgery department. So they already have at least 2 neuronavigation units, which are needed for our sauna biopsy procedure. And I'll break down the gross annual revenue on the next slide of 192 million. Then we can scale up to the serviceable available market where there are 3,000 hospitals that are likely to purchase a neuronavigation system. So they may purchase 1 sauna biopsy unit per hospital, but they'll still have the same consumable costs. Then there's the total available market, which includes the roughly 6,000 hospitals in the US. And these hospitals can be clinics that broadly diagnose and treat brain diseases. We'll see income from 3 sources. First, there's the capital equipment, which includes the focused ultrasound device and the hardware necessary to integrate with the neuronavigation system. And since these hospitals already have a neuronavigation device, they may be willing to purchase 2 or more Sauna biopsy units to accommodate more patients and especially repeat treatments. Then there's the maintenance contract, which includes a software license, service warranty, and personnel training. And there are consumables that include single-use materials that are in contact with patients, which include ultrasound gel or a membrane that covers the ultrasound device. Because we're in the preclinical stage right now, we reached out to our local RWB to discuss the translation to clinical trials. And we were notified that there are not major regulatory hurdles because Sauna biopsy is a non-significant device, so an IDE is not required. This is our go-to-market plan. So since the start of Sauna biopsy in 2018, we've published 3 papers that demonstrate the feasibility and safety in small and large animals, and we'll continue to validate this efficacy and safety for the next year under some various research grants, including an NIH R01. We're developing a clinical prototype to be used in our first-in-human clinical trial with 10 patients. But before this trial starts, we plan to submit, do the FDA pre-submission. After the trial, there'll be another pre-submission, and that would be the first potential exit. We plan to submit a de novo application. So the second potential exit will be when we receive this classification. This will then lead to the product launch, which includes the manufacturing of our own customized focused ultrasound device, some sales and distribution. And we're mainly advertising and talking with the neurosurgery and neuro-oncology departments through direct sales and partnership with the neuronavigation companies. So then Sauna biopsy can be sold as an accessory to their systems. And the third potential exit can be at the end of 2026. And so our team includes Dr. Hong Chen, who is a biomedical engineer, and our interim CEO. Dr. Chen has 18 years of experience in developing ultrasound medical instruments. So after the company is formed and we've recruited a full-time CEO with experience in fundraising, Dr. Chen will still stay on as a scientific advisor. Dr. Eric Luthart is the clinical lead, who is a practicing neurosurgeon with a background in biomedical engineering and is a very successful entrepreneur. His expertise and involvement will guide Sauna biopsy into clinical translation. In engineering, I'm the CTO and Lou is the lead engineer. We've been building the prototype clinical device and are acquiring preclinical data to support our claims. So in summary, Sauna biopsy is a non-invasive tool that will transform precision medicine. And we're now reaching out to interested investors, interested collaborators to set up regular discussions where we can showcase our progress updates. And all this is an effort to raise the funds needed for our first-in-human clinical trial. So thank you for your time, and I look forward to answering any of your questions during our discussion.
Well, thanks to both of our speakers again. I want to remind everyone that you can find their contact information on our website if you want to follow up later. Also, I hope to see everyone in June and July, but I also wanted to give some advance notice about a special session that we'll have in August. Imre Toker, who's an entrepreneur and angel investor, will give a talk to the group about how to start a healthcare startup. So if you're more interested in not the specific science that's being created, but in just how to get started, the August session will deal with that. So thanks everyone for being here. We're just about to run out of time unless we wait a couple of minutes for a last question. But again, I want to thank everybody for attending and once again, thank our speakers.